That I have a different brain as my friends should be clear. However, it is not so evident regarding genetically identical twins. Since they share the same DNA, they often look very similar and share many other characteristics. But what about their brains? Is it possible for brains to be identical? And if not, what actually makes me myself? When you request a new identity card, you're always asked to apply your forefinger to scan your fingerprint because it can identify you with maximal accuracy. What I can already reveal is that brains have been shown to also be one part of the human body, which is unique in every single individual, even in identical twins. For example, the brain volume, the functional dominance of one hemisphere over the other, or a cortical thickness are so individual that they can be used for subject identification. So how is this person-to-person variability even possible? It's a matter of common knowledge that our genetic code is a very indeterministic combination of the DNA of both our parents. As a result, we have 1.5 million DNA variations unique to each of us. Furthermore, each gene can make several transcripts. The set of transcripts is a molecular signature, unique to every individual. Besides, gene expression is influenced by epigenetic modifications. Epigenetic modifications are heritable changes in gene expression, patterns that affect the outcome of a chromosome without changing the underlying DNA sequence. Potentially, then some cells express genes from one parent's allele, whereas other cells use the opposite allele. This parental expression bias may change across different brain regions and ages, which adds to the brain's cellular diversity. In addition, timing and location are also highly influencing gene expression. In general, roughly one half of the variance in individual differences in personality can be explained by genetic factors, while the other half originates from environmental influences. Hence, there is some predisposed functionality in the brain, but the power of the so-called neuroplasticity enables the brain to change its functionality and anatomy depending on actions, circumstances, and experiences in the outside world. It even begins with the baby's conditions in the womb. For example, the surrounding sounds or the mother's nutrition and the resulting available nutrients shape the brain even before seeing the light of day. Most of our neurons are generated early in development and stick around for life. However, some are eliminated, and we only keep the ones which are considered as needed. According to the principle, use it or lose it, this happens especially as a little child, but also later in life. Thus, continuously challenging the brain with physical and mental activity or taking antidepressants helps maintain its structure and function. By contrast, stress, depression, aging, and disease can trigger neurons to break down and even die. Hence, every action of a person throughout the day to some extent influences its brain structures so that new neurons are generated. Neurons grow, new projections or connections are strengthened or weakened. Consequently, the brain is constantly changing, which makes it practically impossible to find two identical brains. The brain's variability goes even so far that whole brain regions like the visual cortex in blind subjects can change their purpose in response to changes in the person's environment or skill set. Hence, it is possible that normally visual domains are active when blind persons read with their fingers. Here, it is important to point out that the phenomenon I describe here, called neuronal plasticity, in some respects decreases throughout our life. Thus, there exists a sensitive period during early life when the different cortices become more fixed in their functionalities. This means that if an older person becomes blind, he's less able to adapt and process and transmit useful visual information than a congenital blind person. We already covered the obvious aspects to mention concerning how does it happen that people's brains differ from each other. Our heritable genetic code and throughout experience and action. However, I can tell there's much more to it. First, a very distinctive characteristic of your brain compared to others is how all the folds and creases are arranged. As the brain grows to fit in the skull, it scrunches itself up in a coordinated way. Some of the convolutions are so identifiable across individuals that they have names, like the central gyrus has. These are the parts which have similar ones in other primates. Many other convolutions, though, vary in, for example, depth, length, or course. These regions of more variation in the convolutions are found in the evolutionarily newer, higher-level association areas of the cortex. As a result, the convolutions in everyone's brain are so unique that MRI studies, a type of scan that uses strong magnetic fields and radio waves to produce detailed images of identical twin babies, could show that differences in patterns of cortical folding can be used to tell them apart with 100% accuracy. Furthermore, our brain contains a surprising diversity of DNA. Contrary to common belief, not all the cells in the body contain identical genetic material. In fact, most cells in the brain have changes to their DNA that make each neuron a little different. Actually, scientists presume that there are almost as many unique cell types as neurons in the brain. One explanation are the many kinds of gene mutation. Mutations are permanent changes in the gene's nucleotide sequence. For example, mutations of any sperm or oocyte cell lead to permanent changes in genetic material that are subsequently inherited by the offspring. Organs such as the skin replace their cells daily. The brain's neurons, though, once established in the fetus, for the most part stick around for life. Thus, somatic mutations in neurons affect brain function, behavior, and the probability to develop diseases forever, often since they disrupt a protein. It has been discovered that a neuron starts with around 600 mutations in an infant, and there's about one mutation every 2 weeks. Hence, by the time a neuron is 80 years old, it has about 2,400 of them. These mutations, for example, can occur due to errors during DNA replication or damage during transcription. Moreover, there are transposable elements in the DNA which are known by the name of L1. L1 elements are especially prone to DNA breaks. They can copy and paste themselves throughout the genome, but also insert large chunks of DNA into the genomes of the cells or even delete parts. Actually, about half of our brain cells have large chunks of missing or inserted DNA caused by L1 alone. In summary, the smallest brain variability exists in identical twins. However, differing social and physical environment, nutrition, actions, varying physical and mental challenges, and many mutations are some examples we discussed, which together explain why identical twins, despite sharing genes, often have different personalities even at young ages. As in conclusion, the evolutionary history of the human brain is written in the genome. However, everyone's brain differs from everyone else's even before the moment of birth, and these differences grow with each interaction with the outside world.